All right. So yeah, let's move to our second talk. We have the honor of having Professor Kunihiko Kaneko. He was the founder of Universal Biology Institute in University of Tokyo, and now he's at Copenhagen University. So yeah, please take it away.
Okay. Yeah, thank you very much for the introduction and invitation. And so I'm Kunihiko Kaneko. And briefly, my introduction is I started from nonlinear dynamics, chaos, and spatial temporal chaos, and this kind of, some of these kind of studies. And then, so now working on complex systems biology, that was from 30 years ago, and now I'm working on universal biology. And so maybe I don't have time to discuss universal biology so much. But anyway, my standpoint in universal biology is that generally life system consists of diverse components and maintains itself and can produce itself. And so the guiding principle here is that some kind of macro-micro consistency. So in a cell, there are many at micro level, many molecules. And these molecules sustain themselves and grow and replicate. And then accordingly, cell reproduces. So this kind of macro state, a cell, and micro state molecules need to somehow kind of consistent growth. So by that, we are trying to find some kind of universal properties of a...
Excuse me,
Yes.
Kaneko-san. Sorry, we might not be seeing the slides that you're sharing. So we just see the introduction, like the introduction
Really?
slide. Yeah, can you maybe try resharing or share a different
It's
screen?
strange.
Thanks.
It's strange. Why this does not work? Okay, yeah. Yeah, actually that happened some time ago, and I don't know why this...
So we see only before
the
you
first, okay.
Yeah.
Before you
So
make the screen, maybe you cannot do not use full screen in the worst option. For us, I think it's big enough.
Okay.
Yeah, here we see the change, and yeah, maybe it's the full screen problem.
Yeah, I see. That's just, or I don't know, maybe. Okay, after I make slideshow and share, maybe that the order may be important. I don't know. So I first, okay, I tried this. And then I have to share. Okay, that's... And I share.
We can see the cursor is moving. Yes.
Yeah, let's see. And I don't know if we do this, you cannot see the second slide.
No, still first slide.
Okay. I don't know how it works.
Oh, now it's moving now.
Yeah, okay. Then maybe I stop slideshow and just this. Then probably that works.
Yeah, it's completely fine.
Yeah, then, yeah. So sorry for the screen is smaller. And I don't know why this happens. Yeah. Okay. Anyway, so, okay. So the brief introduction of myself is I worked on this kind of nonlinear dynamics, and then, yeah, going to universal biology. And then, okay, skip this. So my standpoint in universal biology, as I said, is that there are micro level is molecules and macro level in the cell. And this should be keep some kind of consistency for growth. And according to this kind of consistency principle, I discuss molecule cell level or cell multicellular organism, something like that. And also in the evolution, it's a much different time scales. And so we worked on some kind of cell reproduction versus molecule replication, adaptation, cell differentiation. And recently, we are mostly interested in some kind of evolution, especially evolutionary fluctuation response relationship or some kind of low dimensional structure formed from high dimensional phenotypic space due to robustness and plasticity, and some kind of evolution development congruence. But today, so this is some kind of autocatalysis seminar. So I'm more interested in this kind of reproducing of protocells or origin of life. And then, okay, so especially today, I focus on this kind of waste and sleep. And actually, this kind of sleeping state. So today I talk of this kind of sleeping state of a cell. So it's a kind of dormant state in biology, say. It's some kind of growth arrested state. And so I discussed first about a simple model, and then a little bit more complex model in autocatalytic plus some other reaction network. So the introduction is that most unicellular organisms, such as bacteria, enter to dormant states. So that is, so initially, so if you put some resources into bacteria, the number grows exponentially, and then it stops. This resource is limited and its growth is limited, and then stay there. So the growth, almost no growth state, is last for some very long time interval. So that's called stationary phase or dormant state. And so this is somewhat theoretically interesting because in a system of autocatalytic growth, as you know, this goes exponential growth. But if this autocatalytic process does not work, maybe this growth rate becomes negative, and then it goes to die. But somehow this kind of dormant state means that this growth rate is nearly zero. So in general, staying at nearly zero is not so easy. So then is there some kind of generic mechanism to go to this kind of staying at nearly zero state? And in considering that, so we need to discuss some kind of waste chemicals generally in any cells or protocells. So actually, catalytic molecules are rare in polymer sequence. So if you have just good catalytic molecules, then catalyst can produce itself and you can have autocatalytic set. But there are some kind of other molecules that are created by misreplication process. And then that works as a kind of waste. And waste can maybe inhibit this autocatalytic process. And so this is some kind of waste molecule. So in general, if you have such kind of waste or garbage type molecules, you need some kind of garbage dumping process to keep the growth. And actually, if you have a growth, then as Winston Churchill says, that growth hides every problem. So these garbage components are diluted. And so it can grow itself. But in some sense, that needs some kind of growth and process. But in this kind of sleeping process, maybe this is related to something that this kind of garbage molecules are kind of accumulated. So in this sense, we start a very simple kind of phenomenological model. So that's this in this paper. So we consider just 4 types of molecules. S is that the resource. Resource is coming in from outside of the cell. And then you have some kind of autocatalytic molecule. So good molecule. So active protein. So maybe you can say this is a ribosomal protein or something like that. And so that catalyzes itself and takes some resources by itself. So A is a kind of autocatalytic molecule. But then with some error from this sub-resource substrate, some bad molecule or waste molecules are produced. And so this is A process, and then it's also catalyzed by A. Some bad molecule B is produced. And in general, if the resource condition is good, this process is mostly dominated. But if this resource condition is not good, so these error molecules are accumulated. So the process for this is larger for the good state, but the process for this is lower there. But if the resource is limited, then the process here is larger than this. So that's kind of simple phenomenological model. And then by that, it just the growth decreases by this resource limitation. But here we assume that A plus B makes some kind of complex. So some kind of waste molecules kind of make some kind of complex. So that's also known in biologically. So we have some kind of A plus B goes to C process. So A plus B makes a C. So we have this kind of simple, 4 degrees of freedom ODE. And then by doing this simulation, when this resource condition, so this resource condition is that external resource molecule, where is that? Yeah, so then so this is external, this external condition of external resource is decreased and see what happens. So this is the growth rate of the cell. This growth rate is that so if A works well, so that resource is put in, so injected, and then this cell can grow. So A and B grows, the number of A and B grows. So the growth rate is large for high resource condition. But if the growth resource is limited, then here we found there is a transition to very low growth state. And so actually growth rate is reduced to almost 5-6 digits. So this may be natural. It inhibits the growth and by forming a complex. And then in this region, most active proteins are trapped in a complex molecule. But by being trapped in a complex, then these active molecules are protected from degradation. Because generally in this process, wait. So there is degradation of A. But if A goes to C, and C is complex, so it's less degraded. So it stays there. So it can be some kind of protected. So the good thing for this is that when this resource is supplied again, then the growth is regained. Because in this complex, there is A molecule. So trapped in this complex. A molecules, active molecules are trapped. And then, so when the resource is supplied again, active process is starting to work. And then So from complex, active molecules are regained, and then the growth rate is recovered. And actually, in this case, so to recover the growth, there needs some kind of lag time. And after some lag time, this exponential growth is recovered. And that is also seen in biological cells. And furthermore, this lag time to recover the growth is increased with starvation time. Starvation time is how much this cell is put in a low resource condition. And actually, in this model, this lag time increases to roughly square root of starvation time, like this. And actually, this is some result of some experiments. And it's also showing this. And the reason for this in the present model is that during the starvation, this bad molecule increases in proportion to A because it's produced by, activated by active molecules. But A plus B forms C, and it's balanced. So AB equals roughly C. So it's constant divided by B. So growth of B is roughly inversely proportional to B. So B increases roughly square root T. So to recover the growth, this kind of, these bad molecules should be decreased. And this is in a good resource condition, this is degraded with, reduced by this minus constant. So that means this lambda growth is increases to the starvation time, square root of that. And actually in dynamical systems, this can be understood by something like this kind of two nucleons. And this bad state, final bad states is a fixed point here. But two nucleons are very, so almost tangential. So it takes a very, very long time to go to this. So before reaching this, if you have good, so nutrients are recovered, then it starts to come back. But it takes very long time. So that's basically the story of this simple model. Okay. And then, so we go to kind of, this is very just 4 degrees of freedom system. But now we consider a reaction network. And can we see something similar or generally in this kind of dormant state in this catalytic reaction network? And so we consider a randomly chosen network here. And actually, we previously, long time ago, we worked on some kind of random catalytic reaction network. In this case, Xi plus Xj produces to catalyze by J, Xi goes to Xk. And there's kind of a random reaction network. And we take some kind of nutrient, and by this reaction process, it grows. And in this case, so basically, it exponentially grows. And this in a good state, okay, maybe I skip this. So basically, so actually, these abundances of each chemicals in this reaction network follows some kind of parallel behavior that is consistent with the experiments. And here, so in this case, so again, we consider some kind of transporter. So this resource chemicals is injected into the cell, catalyze this kind of transporter molecule. And in this case, by catalyzing this, and this reaction process works well if the resource condition is good. And in this case, so this balance of concentration of nutrient and catalytic chemicals are balanced, and so that this good exponential growth is sustained there. And so actually in this case, so something like this. But so far in this model, so we worked out about 20 years ago, there is no transition to dormant state. So as long as resource is supplied, maybe the resource is lower, then the growth rate is lower, but there is no transition. So to have transition to growth arrested state, so we consider one more process here. So, okay. It's difficult to search for the process. Okay. So this is the work by Junpei Yamagishi, and that's published this year. So we consider again reaction network. And what is added here is that, so instead of xi plus xj goes to this kind of product, there is complex between. So basically, xi plus xj makes some complex, and then to make some kind of product. So there is one stage here. And then, so if this process to complex to the final product is very fast, then the model is reduced to the problem of this previous model without the complex. But if this process is not so fast, then what happens? And again, if we decrease the resource, and then when this process is not so fast, by decreasing the nutrient supply, then there is again transition. So this growth rate, so sorry for this smaller picture, but anyway, this growth rate decreases suddenly at this point. So here, this point. And actually there is kind of, hysteresis. So it's going down here, it's something like that, and then going up, something like this. So anyway, there is a transition. And actually, in this case, so transition to dormant state, in this reaction network, chemical components are somehow very much diversified. So basically, in a high growth state, so the concentrations are somewhat concentrated on some part of reaction network. And this some part of reaction network works as a kind of autocatalytic module. So this autocatalytic process is working on, focused on this kind of some subset of network. But when the resource is limited, there's some kind of process that chemicals are more diversified to all components. And actually this occurs because in this kind of resource limitation, the process becomes slower, and at many points in the reaction network, some kind of jamming process occurs. And by this jamming, then this autocatalytic process is not working so well, and then it can be diversified to some other parasitic molecules, components. So that's how this kind of transition occurs. So by that, so we have some kind of transition to some kind of high compositional entropy state when this resource is limited. So, okay, basically, if this external nutrient is very high, this concentration is high, it's focused on some kind of autocatalytic subset. But if this resource is limited, then the reaction process is jammed and so stagnated there. And then some other part, so this flow to some other parasitic molecules are more. And then somehow this growth rate is limited and stops again. So that's the basic mechanism here. And actually we can discuss this kind of in a simple, very simple kind of mean field model of this reaction network. So we can analyze this kind of transition in dynamical systems, and this shows this transition is understood as a cusp bifurcation in dynamical systems. And then, okay, we can again see some kind of this kind of lag time starvation relationship that I mentioned previously. So that is somewhat consistent with the experimentally observed dormancy transitions. So, okay. And that is true in this case. And okay, so in the beginning, I said that in the evolution, phenotypic state is somehow dimensionally reduced. So in a good state with exponential growth, so the phenotype has some kind of dimensional reduction. So if you have many, many components, but it's somehow reduced to explained by some subset of molecules, and that describe the growth. But so in this case, so in this present model, so chemicals are concentrated mostly on autocatalytic sets. But of course, there are some other molecules, but this is mostly dominated by these autocatalytic sets. But when the resource is limited, then it's more diversified to some other parasitic subsidiary molecule sets. And then many high dimensional, or so this is out of the dimensional reduction. So we may say that in this case, so diverse components exist depending on many ways, depending on by cells. So we may say this is some kind of Anna Karenina-like phenomena because happy families are all alike. Every unhappy family is unhappy in its own way. So maybe happy cells are all alike, but unhappy cells are unhappy in their own ways. So this is some kind of this kind of autocatalytic by versus many other parasitic molecule sets. So in that sense, so this transition seems to be rather general. And actually, Junpei discussed many cases with other, complex reaction network and or some kind of more realistic network extracted from the real E. coli cells or something like that. So that seems to be rather generic in reaction network consisting of autocatalytic parts and some other parts. So that's basically my talk is something like that. So the first part is with Yusuke Himeoka, and the latter part is by Junpei Yamagishi. Okay, thank you. Sorry for the slideshow does not work. Yeah.
Thank you very much.